don't know what we're waiting for. Alexandra said, give me a minute, so I'm giving her a minute. Okay, great. Well, it is my pleasure to introduce Stefano Anzellotti to the Simons Colloquium. So Stefano was a grad student at Harvard when I met him with Alfonso Caramazza, and did their really beautiful work pulling apart the representation of faces in different brain regions of humans, so where we identify identity versus face parts and also multimodal identity. And he has always had a much bigger, so his work is specific and rigorous, but always connected to a bigger picture and a bigger vision. And so when he came to be a postdoc in my lab, he wanted to solve a problem that seems potentially impossible with fMRI, which is by then, techniques existed to characterize representations in fMRI from their geometry, their static geometry. So you could say in a given region, the pattern of similarity represents, for example, a person's identity. The pattern is more similar when it's the same person, regardless of the image, or image properties, right? It's more similar for two faces pointing left than two faces pointing right, regardless of identity. So we knew how to do that. But Stefano pointed out that that is not the interesting part. The interesting part is how information is transformed as it goes from one brain region to another. So how a brain region's representation inherits from but changes the information that it received. That's the kind of thing that we knew neurophysiologists could study, but it seemed to most people fMRI could not approach, because that's about fast dynamics fMRI does not have access to. And so Stefano invented a whole method and toolkit for looking at how the pattern similarity in one brain region can be understood as dependent on the pattern similarity in the brain regions that feed to it in a nonlinear dependency. This is incredibly hard to figure out how to do with the kind of data that we have. And since he left my lab, he's developed this into a much bigger and more influential toolkit. It's now available on his lab website as a toolkit you can just download and use in your own data. And they have applied it to a bunch of interesting data sets to discover cool properties, representational properties of brain regions in human brains that otherwise seemed impossible to access. So that is an amazing research program on the representation of social information in typical brains. And the project he's going to talk to us today has nothing to do with it, because like many incredibly creative and productive people, he had always a second interest, which is in understanding social deficits in the brain. And so today, and this was another problem I thought was impossible. There's a theme here, which is that in all the work that I did for a decade on human fMRI and autism, we couldn't find reliable signatures of autism, or at least the things we found were tiny and they were buried in a ton of noise. And it started to seem like the sample sizes you would need to study the neural basis of autism in humans would be in the tens of thousands, things that were just not accessible to a single lab or a single, imaging institution. We'd be scanning all day and all night for the rest of our lives. So it seemed like it was just going to be impossible to study with fMRI the signal related to autism buried in all of the noise of those data. And so again, with characteristic creativity, bravery, and technical acumen, Stefano has invented a solution, which I think is really exciting, and that's what we're going to hear about today. So it's my great pleasure to welcome Stefano. Congratulations.
Thanks so much, Rebecca, for the wonderful introduction. It's my pleasure to have the opportunity to share my work, my recent work with you today. So my laboratory investigates social perception, social cognition, and their impairments. In social perception, we are studying the recognition of faces and of actions using neural measures as well as deep neural network models. In social cognition, we are interested in understanding how people make sense and predict the behavior of other agents using deep reinforcement learning and inverse reinforcement learning. But today I'm going to talk to you about another line of work in my laboratory on impairments of social cognition, and in particular on autism spectrum disorder. And this is work largely done by a postdoctoral fellow in my laboratory, Aida Saglinskas. So autism spectrum disorder is characterized by social impairments, communication difficulties, as well as restricted and repetitive behaviors. It is highly prevalent, and it has a very high social cost. So first, I'm going to introduce the problem of individual variation within autism and its implications for the field. Then I will talk about some work we have done to study individual variation in neuroanatomy within autism. And then in the end, if there is time, I'm going to talk about some more recent work we are doing to move towards studying individual variation also in functional responses. Autism is extremely heterogeneous. Individuals diagnosed with autism can have mutations in one of many dozens different genes. In addition, environmental factors and gene-environment interactions further contribute to individual variation within autism. This can lead to failures to replicate. In addition, different individuals diagnosed with autism might therefore need different kinds of interventions. To tackle these challenges, we need to study and understand individual variation within autism. So our goal is to model individual variation within autism in order to hopefully improve diagnosis and to support the development of personalized interventions. When we look at individual variation, this variation can occur at multiple different levels of analysis. There's variation at the level of the genes and variation at the level of environment. And these jointly drive variation at the level of brain structure and function, and in turn, variation at the level of behavior. Today, I'm going to focus on variation at the level of brain structure and function and how they relate to behavior. And we started from studying variation at the level of brain structure because data of structural MRI are a bit less noisy than functional data, and so we decided it would be a good place to start. So when we think about individual variation within autism, we realized we had to deal with another challenge, which is that, yes, the brains of different individuals diagnosed with autism are all different from each other, but also the brains of individuals who are not diagnosed with autism are all different from each other too. And as a consequence, if we look at variation among a set of participants diagnosed with autism, we're going to see differences in their brains, but we will not know which of these differences are actually related to autism versus which other differences are not related to it at all. So I wanted to illustrate this point with an example. So you can see here a set of colored dots. Can you identify any patterns? You can raise your hand if you can see any pattern. Okay, Rebecca, you can see some pattern. What is the pattern? Awesome, yes, right. So you have cool colors on the left side and warm colors on the right side, right? But the fact that there is a lot of variation also within each set of dots makes it harder to identify this dimension of variation between the two sets. Okay, so similarly, if we want to understand individual variation within autism, we need to be able to disentangle the variation that is related to autism from the variation which is in common with the general population. And of course, this idea is not completely new to the field. Researchers have been trying to address this problem to some extent, for example, through the use of case control matching, in which data from participants diagnosed with ASD may be compared to data sets of matched control participants that have similar age and gender and IQ. The challenge here, though, is that there are many, many factors that can affect brain function and structure other than age, gender, and IQ, and often we might not even know what many of those factors are. So ideally, what we would like is some data-driven approach that enables us to discover these kind of factors and separate out individual variation that is related to ASD. Okay, so how can we do this? How can we separate this ASD-specific variation from shared variation? And so I'm going to illustrate the idea of the strategy that we used first with some example that is more intuitive to visualize, and then we're going to apply the same kind of logic to the analysis of anatomical data from ASD participants. So consider the set of images. We have several different pictures here. Some of them with an outline in blue only show different backgrounds. But there's some other ones with an outline in green that vary both in terms of their background, but also they have some additional salient properties that vary. In this case, different handwritten digits that are superimposed on top of the backgrounds. Here, the idea is that, by analogy with the case of autism, there is some variation which is shared by all of the pictures, just as like we may have variation in neuroanatomy that is in common to both ASD participants and controls. But then there's going to also be some dimensions of variation that are unique to a subset of our inputs. In this case, just the images with a green contour. In the case of autism, just the ASD participants. And so, in order to try and separate these different sources of variation, we used recent tools from deep learning and a kind of model. It's called a deep variational autoencoder. And the way it works is we have some picture come in, say that we have a picture that has both the background and then a superimposed digit. And then we're going to have a neural network that is going to infer two vectors of means and standard deviations. At this point, what we're going to do is that for each mean and standard deviation, we're going to use them to define a normal distribution with the mean and standard deviation, and we're going to sample a value from the distribution. We're going to keep that value there in a vector for later. So we're going to do this for all of the different dimensions of the means and standard deviations that we have, and this is going to give us overall latent sample that we call ZJ, so just as an output from those means and standard deviations from that left branch of the neural network. Then we're going to follow exactly the same kind of procedure to then extract a sample ZK from the other branch of the neural network. And then from this, we're going to then finally reconstruct the original image using this kind of latent representations. All right, and so to train this model, we use a loss function. We try to minimize a loss function that has two components. One component is a mean square error loss. This one calculates how different is the reconstruction from the original image. And then we have another component, which is a component that penalizes a difference from a prior that has 0 mean and 1 standard deviation. And this serves as a regularization term. Okay, so we have this network. It takes as input these images, maps it to a set of latent representations, reconstructs the original image. But how does that help us to study autism and separate out individual variation that is specific to it? Well, the key idea is that here we're going to do something slightly different when our input only has the background image and it doesn't have the digit superimposed on it. Now, in that case, we're going to only use one branch of the neural network to reconstruct that input. Right? So in this case, we have one branch, the one in blue, which is used for both images that have background only and background plus the superimposed digits. And this branch is going to learn features that are shared by both kinds of input. By contrast, we have another branch, the branch here shown in green, which is only used to reconstruct the images when they have a superimposed digit on top. And so this branch is going to learn features that are specific to the images that have an image superimposed on top. Okay. Great, so this kind of architecture is called a contrastive variational autoencoder. And what we can do once we have this is we can try to generate images from the latent representation using just the salient space and removing the background. And it can separate out just the digits, removing the background from the images. Okay, so now what we want to do, of course, is not to process images, but we want to apply this to anatomies of participants. And we're going to do something very similar, where when we have the brain volumes of brain anatomies from ASD participants, we're going to reconstruct them using both branches of the network. And when we have brain anatomies that come from control participants, we're going to only reconstruct them only using one branch. And so we tried this first in simulations to check that it would work in a case where we know what the right answer is supposed to be. And what we see is that as we train the network, the loss goes down. That's a good sign that the model is training properly. Then over time, we're better and better at reconstructing the inputs. And critically now, given that these are simulations in which we know what the right answer is, we can ask whether the features of anatomy that are supposed to be ASD specific, that are learned by the model, actually do correlate with ASD specific variation, because we simulated in this case. And what happens to the correlation between those properties of anatomy that are not ASD specific, that are shared, and the ground truth for the properties that we know are, for the variation that we know is ASD specific. And so what we see is pretty much what we'd hoped. That is what you expect. That over training, over time, you see an increasing correlation between the ground truth of the specific variation and the specific variation along the properties learned by the model. Instead, the properties that are shared that are learned by the model correlate less and less with the ground truth of the individual variation of specific properties. So you can see that over training, the network is learning to separate the individual variation along these specific properties versus the variation along the shared properties. Okay, but this is just a simulation, right? And so ultimately what we want to know is how this works in real data. And in particular, what we wanted to ask is, does this process actually help us to reveal relationships between individual differences in brain anatomy and individual differences in behavior that would be otherwise hidden, right? Because if individual differences in anatomy overall just make some variation which is ASD related and some variation which is in common to the general population, now this overall similarity in anatomy might not correlate very well with the similarity based on behavioral symptoms. Instead, our idea was that if we could separate out what aspects of that variation in anatomy are actually related to autism, this now might correlate more with individual variation in the behavioral symptoms. Okay, so we did this using a dataset which is iBite1. It has a little over 1,000 participants collected across 17 different scanning sites. We used a standard preprocessing, and then we applied our CVAE model. Now, one thing that we also wanted to do was to really test whether the key ingredient for this to work is really this kind of contrastive architecture. And so to test that, we also used a control model as a comparison that has the same exact sort of architecture as our CVAE model, except that it doesn't have this trick of using just one stream for the control participants and both streams for ASD participants. This control model instead uses both of the streams all the time for reconstructing the images. And so now we can go and look at the latent representations that are learned by these models and ask how do they relate to individual differences in behavioral symptoms, where ideally what we hope to find is that the properties that are specific to ASD, the ones in the green stream there, are going to particularly correlate with individual variation in behavior. Okay, so to do this, we calculate similarity between all pairs of participants based on the anatomical features, one time for each of the different models. And then we will calculate similarity between participants based on other kinds of properties, for example, their ADOS scores or their DSM-4 diagnosis, as well as other properties. And then ultimately, we can correlate the similarity matrices to evaluate their correspondence. So let's look at some of the results. So here we're looking at the correlation between the different properties and ADOS scores. And first of all, here I'm showing you the correlation between similarity in neuroanatomy and ADOS scores when you look at the control model, right? This is the model that does not separate the specific features from the shared features. And you can see that it doesn't really correlate much with ADOS scores at all. Now, when you look at this for the background features, the ones shared with the general population, versus the salient features, the ones that are specific for autism, you now see selectively a correlation between similarity along the anatomical features that are related to ASD and similarity in the ADOS scores. Now, we can do the same kind of analysis for DSM-4 diagnosis, and we get a very similar kind of result. We have a relationship when we look at the control model, and then much stronger correlation for the ASD-specific features of anatomy. Now, conversely, we would also expect that there's other properties of participants that are shared by both groups that should correlate more with the background features. For example, age and gender. And when we look at that, actually, this is what we find, is that both individual variation in age and individual variation in gender correlate more with individual differences in the anatomical properties that are shared with the general population than with the ones that are specific for ASD participants. So this is basically showing what we're hoping to find, is that by doing this process of separating out individual differences in anatomy that are specific to autism from the ones that are shared with the general population, this reveals relationships between individual variation in anatomy and individual variations in symptoms that was not otherwise always visible. So another question that we wanted to ask about was what happens to effects of motion. We know, of course, that motion is a more severe challenge in the context of functional imaging, but it does affect structural images a little bit as well. And so we wanted to quantify using this Shannon entropy criterion measure, and we computed similarity between participants based on this Shannon entropy criterion that is a measure capturing the motion effects in the anatomical images. Now we compare this to similarity between participants in the control model as well as in our two models that are from the contrastive network, the one that is based on the features that are shared and the ones that are based on the features that are ASD specific. And so what we find is we find some correlation of the control model with the motion effects, but when we look separately for the specific versus shared features, we see that there's a much higher correlation between these effects of motions and features that are shared across the two groups, and much weaker correlation with the features that are specific to ASD, suggesting that also in this case, this kind of strategy is actually also helping us to some extent to remove noise that is in common to both groups of participants. So at this point, one thing that we really wanted to do was to do a test with a new different data set, because one of the challenges in autism research, and that's something that I really learned doing my postdoc, is that sometimes you get some exciting results with a set of participants, and then you try to run it again on a new set of participants, and you get different results, right? And so what we did is we froze the model, we just trained it with abide, and we didn't change the weights at all. And now we started using as inputs a new set of data that was from a new data set, this SPAR VIP data set. That includes a set of participants with 16p deletion, a set of participants with 16p duplication, and a set of control participants. And one reason why we chose this data set is because it also has some genetic information, and we're curious about looking at the relationship between individual variation in the genetic information and individual variation in the ASD-related anatomical features. Okay, so first, we check the same kind of analysis that we were doing before. If we try to look at scanner type, for example, similarity in scanner type, it correlates much more with the shared features and not with the ASD-specific features. Age, again, correlates much more with the shared features, not with the ASD-specific features. And, for example, the DSM-4 diagnosis correlates more with the salient features than with the ASD-specific features, also in the new data set. And then when we look at genotype, actually differences in genotype correlate with individual variation in the ASD-related features, but not with individual variation in the shared features. Okay, so then, yes, we did find that ASD-specific features correlate more with the behavioral symptoms of ASD than shared features, and vice versa, properties that are in common to both ASD participants and controls correlated more with individual differences in the shared anatomical features. So at this point, we wanted to investigate with these new tools a question that has been important in the field, and that is whether there are distinct subtypes of ASD, right? And so we wanted to use this new anatomical data that we have to now ask, when we look at these ASD-specific features of anatomical variation, is there evidence for distinct subtypes of autism? And before we started doing that, we wanted to check that the method is able to do this. So we tried doing some simulations. So we created a simulated set of control participants and two different simulated sets of patients with different kinds of anatomical variation. And so we have these two different simulated subtypes. And first, we're going to look at how these subtypes are distributed in the space of shared features. And then we're going to look at how they're distributed in the space of ASD-specific features. Okay, so in the background space, this is what we see. We'll have a lot of overlap between the two different subtypes. But then when we go and look at the space of salient features, you can see that the space of salient feature nicely separates between the two different subtypes. So this indicates that, in principle, the method has the potential to distinguish between different subtypes within the space of salient features. So next, of course, we wanted to apply this to our data set of real participants in ABIDE. And so we did a similar analysis, but first of all, so we wanted to look at the distribution of participants in the space of the features of the control model. Right? This is the model that does not separate the ASD-specific features from the shared features. And when we look at this, it looks like there is evidence for a little bit of clustering. You can see a couple of clusters there. But at this point, we don't really know what is driving these clusters. Right? Are these clusters driven by features that are shared with the general population? Or are these clusters instead driven by features that are particularly related to ASD? And so when we went and looked at that, it looked like most of this clustering was actually driven by features that are shared with the general population. And instead, when we looked at the space of ASD-related features, the salient features, we didn't really see much evidence of that. So we actually wanted to quantify that rather than just eyeballing it. So we used Bayesian information criterion to identify the optimal number of clusters. We found that the optimal number of clusters was 3 for the shared features, but actually just 1 for the salient features. Okay, so we didn't find any evidence of there being distinct clusters or subtypes of autism based on neuroanatomy. Of course, it's important to point out this doesn't mean there are no differences within autism. There were distinct dimensions that showed a lot of variation. It's just that that variation wasn't organized into discrete clusters. It's also important to point out that this is based on anatomical data, and functional data or other kinds of data modalities might reveal different aspects of this that aren't visible in anatomical structure. Okay, so at this point, we wanted to see whether we could make our results a bit more interpretable. So we have these features that are related to ASD, but what are they, right? Are they affecting particular brain regions, and in what way, right? So far, our model is a bit of a black box. These features aren't that easy to interpret. So how can we do some kind of analysis that is going to help us to interpret what brain regions are being affected by this kind of variation? And so to do this, we had an idea, and we tried to use our model to create simulated control twin participant for each ASD participant. And the way we did this is we took the brain data from an ASD participant, entered into the model, we obtained the features that are shared with the general population and the features that are particularly related to ASD. And then we set the features related to ASD to 0, and then reconstructed out the brain using just the features that are in common with the general population. And what that gives us is basically a simulation of what the participant's brain would look like if it had exactly the same features that are shared with the general population, but without any of the ASD-related features. And so at this point, using these two different brains, we estimated the deformation field that can morph one brain into the other. And from that, we calculated a vector field. And then we calculated, for example, a first dimension that was associated with concurrent expansion in these regions that you see in red and contraction in the regions that you see in blue. One point that was particularly interesting to us in this case was that it involved expansion here in the inferior frontal gyrus and lingual gyri. And this dimension happened actually to correlate with individual variation in the ADUS communication score. And then we identified another dimension, a second dimension, that was associated with concurrent variation in a set of regions that have been associated in social cognition, like medial prefrontal cortex, precuneus, and anterior temporal lobe, and posterior STS. So in this way, we also identified a way to go from a more kind of black box extraction of features that are related to ASD to being able to identify what sets of brain regions are affected by the variation in these kinds of features. Okay, so far this is what we have regarding brain anatomy. We found that, yes, we were able to identify different dimensions associated with changes in different brain regions. And in the case of the first dimensions, they were correlated with individual differences in communication deficits. The taking points from this part of the talk so far is that we have this ASD-related individual variation that is intermixed with variation that is shared with the general population, and that we can use this method, contrastive variational autoencoders, to disentangle the variation which is ASD-specific from the variation that is shared, revealing the structure of ASD-specific variation and its relationship to behavioral symptoms. And of course, we did this in the particular case of autism, but this approach is quite general, so in principle, it can be also applied to other kinds of psychiatric disorders and neurodegenerative diseases as well. Okay, so next I'm going to talk a little bit about some new work that we're doing to try and move towards investigating these kinds of questions in functional responses. Since I'm going to change a little bit, if you have any questions that you wanted to ask about the first part of the talk, maybe I can take one or two questions about the first part of the talk while it's fresh in your mind, and then we can move to the remaining part. Yes? Yes, so region size. So in this case, so there's multiple different kinds of features that we could look at, right? So far, what we used is this determinant of the Jacobian of a vector field as one measure, and that is a measure of cortical expansion or shrinking in different areas, so the size of a certain area, right? And so in this case, the regions that you see in a figure like this, the regions that are in red and the regions that are in blue, mean that as you go, say, from the left to the right along the first dimension, along the first PC, the regions in red are expanding and the regions in blue are concurrently shrinking. Okay, so it's basically this concerted pattern of changes in the size of the regions as you go from one end to the other of a dimension. Now, this particular measure is looking at this question of expansion or contraction. Yeah. That's right. Yeah, exactly. So it's just a data-driven dimension, which is based on the variation that you see in these maps of expansion and contraction across participants. Yes. We could also be looking at other metrics, right? So I think the question is great because so far we're just looking at a small subset of the kinds of things that we could be looking at, right? So right now we have this one thing that we're looking at, which is expansion or contraction of different regions. But what about cortical thickness? We could look specifically, right, at other measures that look at cortical thickness. Another kind of measures that we could look is the measures like shear or other kinds of transformation that aren't just merely expansion or contraction, right? So this is a sort of like, simple first step into looking at a variety of different kinds of anatomical changes that could be occurring, and using different kinds of metrics of anatomy then could reveal different sorts of effects here. Yes.
What do you input to this analysis? It's not like
yes.
It's like
so we have, there's a few steps, but yes, so the input is anatomical images, just T1 anatomical images, MPRH, right, for ASD participants. Now we use our contrastive variation autoencoder model. So it's going to take as input those images, and it's going to infer two sets of latent properties, one set which is shared with the general population, and one set which is instead particularly related to autism. Now we set the properties that are particularly related to autism to 0, and we reconstruct the brain using only the shared properties. Right? At this point, we have our original brain that has both kinds of properties, and we have this new simulated brain that has the same set of shared properties, but none of the ASD-related properties. At this point, what we want to do is compare these two brains, because it's going to tell us how they differ as far as the ASD-related properties are concerned, right? That is, what kind of difference those ASD-related properties produced. And so we do a normalization that normalizes one brain into the other. And then that tells us, okay, how do I need to morph the control brain to turn it into the ASD brain? And that gives us a deformation field. That's a set of vectors that tell us how do you modify the brain of the control to turn it into the ASD brain. Then at this point, with this deformation field, we can look at many different properties of it. One of these properties that tells us about shrinking or enlargement of regions is this determinant of the Jacobian of this vector field. So we can calculate that, and then what we're going to get is a map that for each region tells us how much did it expand or contract when, in addition to the control, to the shared properties, you also add those ASD-related properties. So what is the effect of the ASD-related properties? And so then we have one of these maps for each participant. This is the comparison between the original anatomy of the participant and the simulated control. Now we have many participants, and so we can look at what are principal components of variation across participants. Right? And these are how we get those dimensions. So that PC1 is the first principal component of the variation in the shrinking and expansion across participants, comparing the control to the ASD brain. And the second PC is the second dimension for that. Yes, that's it. Mm-hmm. Mm-hmm. Yes. That's a great question. So actually, let me show you maybe one more time the structure of the model. So the model looks like this, right? And so what happens is we have a brain volume as input on top, and then it comes through these two branches of a neural network, and it gets mapped to those two sets of latent properties, the mu j and sigma j, for the blue branch and the mu k and sigma k for the green branch. Right? And now I can use those sets of properties to then generate out a sample, Zj and Zk, and then reconstruct the original brain as accurately as possible. Right? So that's how the model is built. So the input is the volume of the brain, the output is also the volume of the brain, and the construction is as accurate as possible. When the participant is diagnosed with ASD, I use both branches. When the participant is a control participant, I use only one branch, only the one in blue. Right? Now, what I'm doing this other analysis here is I'm entering as input the brain of an ASD participant. And so I'm getting the blue branches working, the green branches working. I'm getting a mu j and a sigma j from the blue branch, a mu k and a sigma k from the green branch. Now I'm getting my zj and a zk. But now what I'm going to do is I'm going to remove the zk. I'm going to remove the part which is the features that are related to ASD. And I'm going to reconstruct that brain using only the zj like I was doing for the control participants. So then what I did is now I built out a new brain, which is different from the original brain that I put in as input. It has the same shared features, the same blue part, as the brain that I put in as input, but it doesn't have the green part. And so the idea is that it's a brain that's as similar as possible to the original brain in terms of the shared features, but it lacks the ASD-related features. And so by comparing this to the original ASD brain, I can find out what kind of differences in the anatomy are driven by the presence of, in addition of those ASD-related features. Okay. Here. You mean like as a scatter plot of? That's a great question, and no, that is the plot. So yes, that's a great question. So this plot here is along the first two principal components after the deformation, and this scatter plot here of purple dots are the scatter of participants. So that's a great question, and no, we do not see the clustering, evidence for clustering in the ASD-specific space, even after using this kind of deformation-based analysis. That's a great question. Yeah. All right. Awesome. Then, oh, you have another question. Okay, great. Awesome. Go for it. Mm-hmm. Yes. Of course, yes. So to clarify the subtype analysis, there were two sets of analysis that I did regarding subtypes. One is in a simulation where we on purpose created different subtypes. These are not real data, simulated data. The purpose of that was to demonstrate that if the subtypes are there and are clear, then the method is able to actually see them. Because what if maybe the method doesn't work? So then even if the subtypes are real and there, and then the method doesn't see them, that wouldn't be very interesting, right? So that was the first analysis, to say, let's create a case where we know for sure that there are subtypes because the data is made up, we simulated it, and we built the subtype in. And so in that case, you could see that in the salient space, you could really see the different subtypes to be separated. So there isn't a general problem with the method that it can't see subtypes. In principle, the method has the ability to do this, right? And then the second analysis was, let's see what happens in our real abide data, right? Are there subtypes there? And so in that case, we looked at that first using the anatomy features that were from the control model, so that didn't separate out shared variation from ASD-related variation. In there, it looks like you have some clusters, but we don't know yet whether those clusters are because of properties that are shared with the general population, or whether they are because of ASD-related properties. And so when we separated that out with our contrastive VAE model, that's when you could actually really see that there was some evidence of clustering in the shared features, but not in the features that are particularly related to ASD. And so we could quantify that. So this suggests that we didn't really see, even though in principle the method can see clusters, as we showed in the simulations, in the real data, you only saw clusters in the shared properties, but not as far as the ASD-related properties are concerned. Okay. awesome. Oh, okay, you have also another question. Awesome. No, that's good.
This may be an ignorant question, but I noticed in screen maps the cerebellum is not on there.
Oh, that's a great question. You're right. I just didn't put it in the slides, but we have cerebellar effects in the paper figure as well. So you're absolutely right. There are some effects in the cerebellum as well, and so it's not like cerebellum does nothing. Sorry for people who study the cerebellum. It's just there are effects there. They are in the paper, but I just didn't put them in the slides. Okay. So then, at this point, one of the next things that we're trying to do, I don't know how much more time I have, is 4:45, another 10, 15 minutes? Okay, great, awesome. So one of the next things that we would like to do is to then extend this approach to investigate individual variation also in functional responses, in addition to anatomy. And because we think that functional responses might reveal things that aren't observed in anatomy. For example, it might be that in anatomy we do not see different clusters within the ASD group, but it might be that functional data could show such clusters. So we wanted to start by trying to use functional connectivity. And this, by the way, is work done together with a brilliant undergraduate in the lab who is now actually a graduate student at Brown. The first thing that we realized when we tried studying individual variation in functional data was that we had a huge problem because there is a lot more noise in functional data compared to anatomical data. And if the plan is to average results across many participants, then it's more manageable to handle that kind of noise. But if you want to have the level of reliability within one single participant to be able to study individual variation, that is really, really hard. It's really hard to do. And so the first thing that we thought is, if we have any hope of trying to do this, we'll need to change the way that we denoise the data. Maybe we need some better denoising strategies. And so actually we realized something that was a bit unexpected, which is that we could use a very similar logic and strategy to what we had used to separate ASD-related variation from shared variation to extract signal and separate it from the noise. And the idea here is that we can use a set of regions that we're interested in, like gray matter, where we have a mixture of neural responses and noise. But we can also consider another set of brain regions where we don't really have neural responses, for example, cerebrospinal fluid. Right? These regions are also going to be affected to some extent by noise. And so we can extract time courses of response from our regions that have both signal and noise, and from our regions that have just noise. And then we can play the same exact kind of trick, but with time courses from voxels. So we can then take voxels from regions that we're interested in, like gray matter, that have both signal and noise. We encode them with this contrastive variation autoencoder, has two branches, and reconstruct them out using both. And then we're going to take the voxels from regions that only have noise, like CSF, and we will only use one branch. So there is one branch that's in common to both that is going to encode variation in the noise, and the other branch that we only use for the gray matter voxels that's going to encode information about the signal. And then, yes. Yes, this particular analysis was then done with resting state, but the same kind of approach could be used for task-driven data. I don't see any problems with that. Okay, and then in the end, for denoising, what we can do is we can take our input data, and then it's going to get mapped to both sets of features, the one in common to both noisy, to gray matter and CSF, and the ones that are just specifically for the gray matter. But then we can set to 0 all the noise features, and we can just reconstruct the signal out using only the branch that encodes the variation that's specific to the signal. Okay, so how well does this work? Well, first of all, we wanted to try and test it out using simple simulations where we knew what the right answer is supposed to be. So we created some ground truth of the signal, some ground truth of the noise, then we mixed them, created simulated observations, and then we denoised the data. We used different approaches. We used this new method that we call DeepCore, and we compared it to CompCore, which is another very popular method that uses a bit of a similar logic, but largely relying on linear models. That's quite effective and quite popular today. And so we can compare now the ground truth to the denoised data using CompCore and then using the new method DeepCore. So we are going to do it at different levels of noise. So here's with a bit less noise. What you see in blue is the ground truth. In orange, you see the denoise data. You still see a little bit of the pattern of the noise in the denoise data with CompCore. And if you increase the amount of noise, you can actually see quite a bit more of the noise pattern, of the sinusoidal noise pattern in the orange data. Now, if we use DeepCore for the same data set, this is the kind of results that we get. And so you can already sort of get an intuitive idea when you look at this that the answer in the case of DeepCore is much, much closer to the ground truth. Now, of course, we wanted to do this also with real data. And so the problem with real data is that we don't really know what the right answer or the ground truth of the signal is. But then what we did is we used the fact that there's networks of brain regions that show high functional connectivity for regions that are within the same network and low connectivity for regions that are in different networks. And our reasoning was that if there is noise that is affecting the whole brain, you could overall inflate correlations between brain regions, affecting both within regions that are within the same network, but also regions that are between. And so this is the kind of correlations that you see within network without any denoising, so very high. Between network without any denoising, that's still quite high. And then here I'm going to plot the within minus between, the difference in correlation within minus between networks. Now, this is what you get with CompCore, right? So it's good. It looks like it's working. It's reducing a lot of these kind of correlations that might be driven by noise. The correlations for between are a lot lower. The ones for within also get lower. And now the difference within versus between is even a bit larger. Now, if you use DeepCore, what actually happens is that you increase the correlations within network, but further decrease the correlations between different networks, and you further enhance the difference of within minus between correlations, suggesting that also in the case of real data, this method might be doing a better job at removing noise. Now, finally, to have a clear indication of this, we wanted to run another set of analysis. And this is another set of simulations, but with more realistic data. So this was a case where we wanted to say, let's see, we want to analyze a case where we really know what the ground truth is, but where the simulated data are a lot more plausible than what we used in the first set of simulations. So we used a package called Brainiac that can generate event-related responses and mix them with multiple kinds of realistic fMRI types of noise that are extracted from real fMRI data and estimated from real fMRI data. So we created a simulated data set with responses to different kinds of events. We added multiple different types of fMRI noise that were extracted from real data. And then this is an example of voxel denoised with CompCore. In blue, again, you see the ground truth, and in orange, the denoised data. This is the kind of effects that you get with DeepCore. So you can see there that the answer obtained by DeepCore in orange is much closer, and the R-square is much higher. Of course, this is one representative voxel. So if you look and average the results and the R-square across all the different voxels, this is the kind of result that you get. So you can see that the average R-square for voxels denoised with CompCore is just a little under 0.2, and the average R-squared for voxels denoised with DeepCore is around 0.6. So it's about a 400% improvement in the R-square between the denoised data and the ground truth that is obtained by DeepCore. Okay, so this is where we are so far. Is this good enough in order to see individual variation in functional data between different ASD participants? We don't know that yet. That is something we're trying to find out right now. We're working on it right now. We hope it's going to be good enough. If not, at least hopefully this denoising approach is going to be helpful for other applications as well. So, maybe at some point in the future, I'll be able to tell you whether this was sufficient to see this individual variation at the level of functional data. So, next steps, of course, first of all, studying individual variation in functional measures, taking advantage of these new denoising methods we developed. Second is going to be trying to integrate more different kinds of data modalities. As we said, anatomical data only give us one window into these kinds of differences. Functional data gives us another, but ideally we will also be able to integrate diffusion-weighted images as well as potentially methods that have faster temporal resolution like EEG or MEG. And ideally, in the long run, potentially also genetics data. And then ultimately, one thing that I'm interested in is thinking about studying comorbidity using these kinds of approaches, because I think of comorbidity as being very closely related to the problem of individual variation, just as a sort of like slightly larger scale, where you have variation across multiple different kinds of disorders and then within individual disorders. And so I think that ultimately what we'll need is models of individual variation that operate not only within a single disorder, but across multiple disorders. Right. Thank you. I think this is all that I was planning to talk about today. I wanted to thank, of course, Idis, who did a lot of the heavy lifting for this project, Josh Hartson, who collaborated on the anatomy project, and Yuju, who worked really hard on the denoising project. Thank you very much. Yes.
I have a quick
Awesome.
question.
Yes.
I mean, for the second part, I saw the denoising method is working on the chronic and drifter noise. I was wondering, is it also work for the suddenly motion noise?
Yes. Will it also work for, I see, I think I understand your question. Let me see if I get it correctly. So, because I plotted this slide, right, that shows these different kinds of noise, right? And this noise includes physiological, drift, or regressive task, but it doesn't have head motion, right? And so are you asking, does it also work for head motion?
Yeah, I mean, its noise is very chronic, right? Like,
yes.
If a motion noise happens, it will suddenly a bigger noise.
Yes. So this particular software package doesn't allow for simulating that kind of noise, but that's why we also wanted to look at the effects on real data, where it does seem to work better. My expectation is that, yes, it should be able to capture sudden noise, because if you think about it, what it's trying to do is to identify features. So it's going to have some time courses that are from a voxel, say, in gray matter, and a time course, say, from a voxel that is in cerebrospinal fluid. And then this method is identifying features that are shared by those two time courses versus features that are unique to the time courses that are in the gray matter voxel. So if you had a rapid sudden motion, that would lead to a spike in the gray matter voxel, but also to a spike in the CSF as well at the same time, right? And so that particular spike, it should end up as part of the features that are shared by the CSF voxels as well as the gray matter voxels, and not to the unique features. Yeah. So, but I think it's a really good question. It could be cool to run some specific tests to really probe that particular kind of motion and say, what if we put in data set that has this particular kinds of spikes? I mean, you can get a bit of a sense, from the kind of reconstructions that you don't really have these big spikes in the reconstructions or issues with that. But yeah, I think it's a really good question. It'd be interesting to look specifically at that type of noise. Yeah, great.
Going back to the first part, I'm curious, like when you train the model and then you generalize with Safari data set,
yes.
To what degree can you predict whether the brain you're looking at is from a control or from someone with ASD?
Oh, that's a really good question. So we haven't really tried that. So this model is built more to model variation within each group rather than to model distinctions between ASD versus control. There are other studies that have done that kind of thing, like train a deep neural network to separate ASD participants from controls. They get some reasonable accuracies. They're not perfect, but they do okay. Yeah, so it's possible in principle to train models that get a reasonable amount of chance accuracy to distinguish ASD anatomies from controls. But we haven't tried with this particular model because the focus was really on trying to study individual variation within the ASD group rather than differentiating the ASD versus controls. Yeah. Yes?
Can I ask you a few technical questions about this slide?
Of course.
I don't understand, what are the dots?
Okay, so the dots are different samples from the latent space of the model. So in the model, you can sample every time because it's this generative probabilistic model, right? So you can sample the sample Z's, and you're going to get some value for each participant. So we want to make sure that these things are very stable across different samples. It's not that just we got lucky once, we got the right sample. So you can run this several different times and test whether you find this effect consistently across multiple distinct samplings from the latent space of the CVA. Yes.
And how should we think in general about the fact that in this part of your talk, the effect sizes are tiny? There's a lot of dimensions of variation, right? And their relationship with symptoms
I wouldn't expect it to be very, very strong, right? Because so these measures, this similarity between the anatomical properties is capturing overall similarity in any kind of ASD-related variation that you have in the brain, but potentially only a small subset of that might be related to a particular cognitive or behavioral measures. So ideally what we'd want to have, I think, next is to have a set of much more fine-grained behavioral measures. I think that's a major limitation for future steps. So, for example, here we're comparing things to measures like the ADAS questionnaire, right, or DSM-4. These are, I think, relatively coarse characterizations of individual differences in behavior. So I think to make these effects be stronger, what we really need is much more comprehensive characterization of behavior and individual variation in behavior, and then start studying, which kinds of individual variation in what aspects of behavior are related with what subset of the anatomical variation that we see. When we just compare similarity in overall variation in any aspect of anatomy that is possibly related to ASD, with one rather specific limited behavioral measure, then that's when you're not going to see extremely high correlations. That is my expectation. Yeah, that's a great question. All right. Any other questions?
Any way of getting questions from the online? No, we have no way. They have to just hope we ask their questions.
Thank you.
Okay.
If anyone is watching this online and has questions, you can send me an email.